Hello everyone. So from today we are starting with the photosynthesis part, and we are starting plant physiology today. From today, so firstly, the reaction of photosynthesis is this, okay? And the oxygen which released during the photosynthesis comes from H2O, comes from water, not from carbon dioxide. Okay, so the structure of chloroplast is very basic, and we will discuss later also, like what is F1, F0 particles, perimitochondrial space, outer membrane, inner membrane, because this is very basic. So we will discuss it later. What the thing you need to remember today, that chloroplast is a site of coupled oxidation reduction reaction. So if the question comes that which is the site of coupled oxidation reaction, so you have to write that chloroplast is a site of coupled oxidation reduction reaction. Okay, so this is clear. Now let's come to the pigment part, that what are the pigments that are involved in the process of photosynthesis. First we have chlorophyll. Okay, chlorophyll, we have chlorophyll A and chlorophyll B. The formula of chlorophyll A is C55H72O5N4 Mg. The chlorophyll A is the main reaction center, and other than chlorophyll A, all the pigments are known as accessory pigments, or what we call antenna pigments. Okay, now chlorophyll A has methyl group and chlorophyll B has aldehyde group. What you need to learn here is that in both chlorophyll A and chlorophyll B, magnesium is present. Okay, the next pigment we have is carotenoids. Okay, carotenoids, we have carotene and basically it is of two type, carotene and xanthophyll. Okay, carotenoids are both scavenger as well as quencher. Okay, now pollen grains, know that in the pollen grains sporopollenin is present. The sporopollenin is also made up of carotene. Carotene is one of the type of carotenoid. Okay, so the sporopollenin is made up of carotene. Sporopollenin. Okay, so sporopollenin prevent pollen grains from any damage, like physical, chemical, biological. It prevents the pollen grain from any of these damages. Okay, I think the two pigments we have learned is right now, like chlorophyll and carotenoids. Now we have phycobilin. Okay, so phycobilin is one of the water soluble pigments. It is of two type, that is phycocyanin and phycoerythrin. Phycocyanin is purple in color and phycoerythrin is red in color. Okay, so the bile salt composition is same as a phycobilin composition. Okay, the bile salt composition is same as the phycobilin composition. Okay, okay, now very important thing, very important this is. Now look at this is the stroma part and this is the thylakoid. Okay, in the thylakoid a reaction take place in which two molecules of water break down into 4 H plus ions, 4 electrons and oxygen molecule. Okay, now the 4 molecules that are released in this particular thylakoid goes to the PS system second. Okay, 1 electrons, like if it takes 1 electron, okay, so 1 electrons lead to the influx of 2 H plus ions. Now 4 electrons are going to pH, like PS second. So in return of these 4 electrons, 8 H plus ions influxed inside the thylakoid. Okay, 1 electron is equals to 2 H plus ions, so 4 electrons is equals to, we have 8 H plus ions in this. Now 4 H plus ions are already present and 8 H plus ions get influxed. So in total the number of H plus ions in thylakoid is 12. Okay, then the cyclic reaction take place, then basically the light reaction take place. The light reaction which will be discussed in detail in later, but the light reaction leads to the formation of ATP and NADPH. Okay, ATP and NADPH, and the both ATP and NADPH gets consumed during the dark reaction. Okay, it consumed during the dark reaction, which is like C3 C4. Okay, mainly to C3 ही होती है, like C3 is the main one, and other than the C3 there is CAM cycle, there is C4 pathways. Okay, we will discuss also. Okay, so this is the C3 cycle example in which carbon dioxide leads to the formation of 3-phosphoglyceraldehyde, which later forms glucose, and then it further forms sucrose. Okay. Now come to light reaction. Light reaction is of two types, that is photophosphorylation, and which is of two types, cyclic and non-cyclic, which is of two types like cyclic and non-cyclic. Non-cyclic photophosphorylation is also known as Z scheme. Okay, it is also known as Z scheme. Firstly, we will discuss about the non-cyclic pathway. The non-cyclic pathway takes place in thylakoid. It will not take place in stroma. It will take place in thylakoid. Okay. As we go down, the reduction potential will get increase. Okay. This reaction takes place inside the thylakoid. Now the electrons first goes to PS second. Okay, then the PS second is also known as P680, and the PS1 is also known as P700. So the electrons will firstly go to the PS second. It will get excite, and then it will get transferred to pheophytin, then plastoquinine A, plastoquinine B, cytochrome B6F, then plastocyanine. In the cytochrome B6F, 2H plus ions also come from stroma to lumen. Okay, then PS1, then A0, A1, FES, FT, FNR. ठीक है, ferredoxin NADP reductase. Okay, look at here. A0, A1, FES is of three type, FEX, A, B, then FT, and then FNR. And it will lead to the conversion of, NADP to NADPH. Okay, so firstly the electrons get donated from PC. You have to learn it. So firstly the electrons go from here to here, then it will take electrons from this reaction. Okay, so firstly the electrons get donated from the P680, then it will take the electrons from the photolysis of water. This reaction is known as the photolysis of water, which takes place in the thylakoid. Okay, now two inhibitors we have to learn here, that paraquat blocks the FNR. This FNR gets blocked by the paraquat, and DCMU, which is an herbicide, that is dichlorodimethylurea, blocks this QB. ठीक है, so dichlorodimethylurea blocks the QB. Okay, so in presence of paraquat, the FNR gets blocked. Because of this, the ATP and NADPH both will not form. Okay, because the photophosphorylation reactions, like the photophosphorylation reaction, lead to the formation of ATP as well as NADPH, because it is non-cyclic pathway na, so it leads to the formation of both ATP and NADPH, and the both ATP and NADPH will get utilized in dark reaction, which take place in stroma, not in thylakoid. Okay, now let's come to the cyclic photophosphorylation. Okay, now firstly you have to remember that it will take place in stroma lamella. You know about stroma lamella? If not, I will tell you about it. Okay, this is the stroma lamella. Okay, which connect the two adjacent granum. Okay, which connect the two adjacent granum. Okay, this is the stroma lamella. So the cyclic photophosphorylation takes place in the stroma lamella. Okay, the stroma lamella characteristic is it will not in the stroma lamella PS second, that is P680, is not present. We will read about it later, that the appressed one, non-appressed. Actually now the stroma lamella is non-appressed one. Okay, and here the PS second is absent, and PS1 and ATP synthesis is present. So PS second is basically absent here. Okay, so cyclic photophosphorylation takes place in the condition when NADPH is too much, but still the plant require the ATP for the other reactions also. Okay, so cyclic photophosphorylation take place in the condition when the plant has lot of NADPH, but it still lacks the ATP. Okay, so this is how this reaction similar, like it is very similar. That's the PS1, A0, A1, FAS, FD, FNR. But the thing is that in the cyclic photophosphorylation only the ATP gets produced, not the NADPH, because the plant already has a lot of NADPH. Okay, now let's here I have written like something like detailed terms like Pheophytin. What is pheophytin? Pheophytin is basically that magnesium in chlorophyll. The magnesium was present in chlorophyll A and B, both in both the things. Chlorophyll, magnesium was present. So in pheophytin na, it is same as chlorophyll, but the magnesium in it replaced by H plus ions. Okay, QA QB, they both are plastoquinine. PC plastocyanine, that is copper, is present here. A0 is the modified chlorophyll. A1 is a phyloquinine, that is vitamin K. Fes FEXAB, ferredoxin, NADP reductase. So you have to learn the name because it is a detailed meaning of all the term that we have discussed in the cyclic and non-cyclic reactions. Okay, this is very important. Okay, DBMIB, 2,5-dibromo 3-methyl 6-isopropyl p-benzoic. It is like a very important question, that it is an inhibitor of cytochrome B6F2PC. You have to remember that we have read till now we have read the 3 inhibitor. The first one is paraquat. The second one is DCMU, and the third one is like paraquat, DCMU, and the third one is 2,5-dibromo 3-methyl 6-isopropyl p-benzoic acid. It blocks the cytochrome B6F2PC. Okay, it blocks this pathway, this cytochrome B6F2 PC. I will recommend you to learn the non-cyclic pathway one, because if you learn the non-cyclic, you will eventually learn the cyclic one. So firstly you have to learn the non-cyclic pathway, then you will remember everything. Okay, you have to imagine this. Okay, like P680 receives only the single electron at one time. Like at one time it will receive only the single electrons. It will not receive 2 electrons or 3, because the 4 electrons release in the stroma na, in the thylakoid na, so it will only take 1 electron at a single time. ठीक है? Why? Because it has that potential only. Okay, now the 3 electrons which are not received by the P680, now the 3 electrons have the capacity to damage the chloroplast because it is present in the thylakoid. But so the plant has done an evolutionary mechanism. It has evolved a special mechanism for it to protect itself from the 3 electrons, that is oxygen evolving complex. That is the oxygen evolving complex. In the oxygen evolving complex, manganese, that is Mn, is present. Mn has 2 characteristics. It has variable valency, like in the chemistry part it has 7 valency. When you are reading about biology, it has 5 variable valencies. The second characteristic of it is in the manganese, the S state mechanism is present. Okay, in the manganese the S state mechanism is present. So 2 terms. Okay, this is very important. D1 protein and D2 protein. PSBA synthesize the D1 protein and PSBB synthesize the D2 protein. What is D1 and D2 protein? You will remember it here. The D1 protein takes electrons from the P680 takes electron from the photolysis of water through this D1 protein. The D1 protein has tyrosine amino acid. Tyrosine has hydroxyl group. You will learn it now in the biochemistry part, that tyrosine has hydroxyl group. So tyrosine hydroxyl group get electrons from these states. So here the photolysis of water taking place. Okay, these electrons are received by S0, S1, S2, S3, S4. These S are the variable valency of manganese present in the thylakoid of the chloroplast. Okay, S0, S1, S2, S3, S4. These are the 5 variable valencies of manganese present in the biology. So the first the electron are received by S0. It give electron to 1, and the 1 electron is received by this. It gives electron. So like this is the mechanism through how the chloroplast protect itself from the other 3 electrons which are not received by the P680 at that point of time. Okay, you have to learn it. D1 and D2 protein. D1 synthesized by PSBA, D2 synthesized by PSBB. Now D1 and D2 protein both are present in the thylakoid membrane. Okay, you have to learn these both of these things, that what are the ions required for the photolysis of water. So these are manganese, calcium, chlorine, and for oxygen evolving center, like manganese, calcium, chlorine, and bicarbonate, that is HCO3 minus ions. Okay, so this is the light reaction is completed here. We will discuss about the dark reaction in the next chapter. Okay, so till now we have completed the basic of photosynthesis. Other than chloroplast structure, we have discussed everything, the basic, the light reaction. And this lecture will be benefited for you if you are a class 11th NEET student, if you are preparing for CSIR net, if you are doing your BSc MSc, if you are doing your BSc botany honors. It will be beneficial for you because I have covered everything in this particular lecture. Other than the chloroplast structure, everything is mentioned in this lecture. Okay, we will discuss the chloroplast structure later. Thank you.